So our next speaker today is Sofonias Tessema from UCSF. Thanks.
Laser forward and back. Laser forward and back.
Okay, thank you very much to allow me to speak about the work that we're doing at UCSF. We're trying to understand very fine scale level of population genetics in the transmission area of Southern Africa, mainly in northern Namibia.
The green one up above it.
Yeah. Okay, so malaria in Namibia is a low transmission area, and Namibia made a very substantial level of success in reducing the number of cases in the last 10-15 years. The number of cases is reduced to a very low level, but there are still pockets of transmission in northern Namibia, mainly concentrated in border regions of southern Angola, where there's a high transmission of malaria. We have two study sites. One is in the Kavango region, and the other one is in the Zambezi region, which is in northern Namibia. In the Kavango region, we had 24 clinics, which are border clinics, and we also have in the Zambezi region 6 clinics, which are distributed in different parts of the region. We have samples from 2015-16 season in the Zambezi region. So we have samples from the Zambezi region from 2015 and 2016, whereas in Kavango we only have samples from 2016. What you see is in 2015 there are few cases, and in 2016 there was an outbreak. There was a very large number of cases within a short period of time. So we analyzed this data to understand fine-scale level of population genetics in northern Namibia. Samples were collected from DBS and RDTs. From the Kavango region, we have RDTs, and we also have stickers which just stick into the RDTs with a lot of information such as age, the primary residence, and if they traveled, where they traveled to, and how many nights they spent in the traveled area. And then we genotyped all these samples using 26 microsatellite markers. What we found in northern Namibia is most of the samples are really polyclonal. You can see here in the y-axis is the multiplicity of infection, and the x-axis is the different clinics where we sampled. So of these 30 clinics, you can see most of the clinics have higher level of multiplicity of infection, and about 80% of the samples in Kavango, even if it's a low transmission area, we do see polyclonal infections. And in Zambezi, relatively lower level of polyclonality, but still there is a high level of diversity in the Zambezi region. So we have this data, and we are interested in understanding mainly the connectivity between infections at different scales in this low transmission area. Which individuals are transmitting infection and where, and which communities are connected to each other. And we can also go to the level where we can see connectivity between districts and connectivity between regions. We're also interested in understanding the scale of transmission. What is the scale of transmission? Even if we have samples which are collected in a very short window of time, but can we see any local propagation of these transmissions? We're also interested in understanding the contribution of imported infection to the local transmission, because this area borders a high transmission area of Angola. So is there a transmission from Angola? Is that contributing to the local transmission in this region? We used the transmission network model, which was described by Brian on Monday, to understand the connectivity between each individuals. So what you see here is every circle represents a sample, and the line which is connecting is the darkness of the line is proportional to the probability of transmission, and the colors in each circle is filled with the location where we collected the sample. These are the clinics, as you can see, from west side of the region to the east side of the region, from blue to red. So just eyeballing these, you can see similar colors are connected to each other more frequently than those samples which are connected from distant areas. But it's interesting, just to draw your attention, you can see here samples collected from very similar clinics connected to each other. But we also see connectivity between samples from about 200 kilometers apart. We can see here it's just to show the connectivity by the distance between the clinics. We have higher level of connectivity at a shorter distance, but we also see connectivity of samples transmission up to 200 kilometers apart, so suggesting that there is local, very focal transmission and also transmission between all these clinics. When we compare this with just randomly generated network, what we see is we don't see that pattern. You can see the gray background is the randomly generated network. So we do see more focal connectivity in this transmission area. We also compared these data using other traditional methods such as genetic differentiation and also distance-based approach. So we tried to use those approaches which enable us to include all the polyclonal samples, because most of our samples are polyclonal. What you see here is we used an IBS distance, identity by state distance, which is described in this paper. What we did is just pairwise relatedness between samples based on their allele sharing, and the y-axis here shows that the proportion of highly related samples, and the x-axis shows the distance between the clinics. When I say highly related, it is genetic relatedness greater than 0.7. So highly related samples are found in very short distance, and the proportion of highly related samples decreases with distance. But using GST, which is an equivalent of FST, using genetic differentiation method, we don't see that pattern. So what we think is this method will not be able to give us the resolution that we want at a really fine scale level. Similar result was recently reported from Thai-Myanmar border, where they don't see association with FST, but they do see association with highly related samples, the proportion of highly related samples. We are also interested at looking at, also we do see a decrease in the relatedness of parasites with distance. Are there areas where we can see a lot of connectivity? Which areas are more connected to each other than others? So we have multiple data. We have human mobility data. We have also the genetic distance, which is determined by IBS, and we have the network distance. And also we can compare this kind of observation using traditional approach such as GST. What we can see is, you see the mobility data, this heat map shows the proportion of time spent in different locations. When they move to different towers, it just detects the proportion of time they spend in that tower. So you see this is the capital of the region, so people travel to the region a lot. And mostly, but other than that, mostly the travel is within short distance. So this might explain what we observe in the transmission network data. So with using IBS and the network-based data, what you see is here is there are regions where we see a higher level of connectivity, but we don't see higher level of connectivity in other regions. So there is a very fine scale level variability between population connectivity in this region. Also, the network-based data shows similar kind of result with the IBS distance. Using this data, we are also able to determine which areas are sources and which areas are sinks. So what we found is there are two clinics, the Shambayu area and this is the district hospital area, which are predicted to be estimated to be the source of many of the infections. And this region is predicted to be the sink where there are more infection coming to this area. This might be interesting because there are new agricultural projects and settlements in this area that might be the cause for importation of malaria into this region. We're also interested in understanding the contribution of imported infection to local transmission, how much genetic information can add on on travel history or epidemiological data to actually and accurately classify imported and local infections. So what you see is the same transmission network but colored differently. In this case, the blue color represents that people who haven't traveled to Angola, and the red color represents those individuals who travel to Angola and spend at least 3 nights in Angola. So we do see connectivity between those individuals, assuming that those who travel to Angola are imported infections. We do see connectivity between imported and local infection, but this is really complex. I will just show you there are 3 different scenarios. You can see an imported infection contributing into local transmission, the one which is circled here, and the other one is you can see connectivity here between just imported infections, and also we do see connectivity between local infections transmitting into Angola. So there's a two-way transmission here. There are samples imported from Angola and transmitted into Namibia, and there are samples which are showing us that transmission is happening from Namibia to Angola. This is a really complex phenomena. So it's the dynamics of these importation in areas where it's mostly border clinics, as you can see on the map. The border is very porous, and people can move around in both directions. So quantifying importation using genetic data, you probably need a lot of information from both sides of the country, and also this is really a complex scenario. But we try to quantify this using just the transmission network. What we see is we do see connectivity between Angola to Angola samples, and we also see connectivity between imported to local infections, and majority of the connections are local to local infection. This result is interesting because the programs were very focused on importations from southern Angola, but there are also local transmission which is happening in the Kavango region. What we estimated based on this transmission network, we estimated higher level of importation than which is captured by travel history. With all the limitation of travel history, how accurate it is, how much complete it is, this information can add on the epidemiological data that we can collect. The other approach that we're taking to understand fine-scale population genetics in northern Namibia is trying to identify geographic origin of infections. We use this approach called SCAT, smoothing and continuous assignments. Assignment tests has been used for mammals to assign populations into different populations. We have 2 approaches here. There's smoothing assignment and there's a continuous assignment. In the smoothing assignment, allele frequencies are estimated at each location, and the probability of each individual genotype for each location is determined. So we will get a probability by which whether that sample belongs to that specific location. So we did a modification because this was designed for mammals and diploid organisms, and we modified it into haploscat. We called it haploscat, where we are able to use it for haploid genomes. So using this approach, we did a bit of validation of this method, and what you see here is we have samples from 3 districts in Kavango and 1 district in Zambezi. These samples were collected from 3 different regions, and the sample size is kind of comparable. So we try to assign samples which are collected from Rundu into either of these 4 locations. And what we found is only 30% of them are assigned to Andara and 30% to Nyangana, but about 40% of them are assigned into Rundu. It's like we have 1 sample, if this sample belongs to any of these locations based on their genotype and allele frequency. So what we found here is 60% of those samples from Zambezi were assigned to Zambezi, whereas 40% of them are assigned to Kavango, suggesting that there is a mixing between these 2 regions, even if there is low transmission in this area. And also this suggests that there is some sort of allele frequency difference which can enable us to do these sort of assignments. To summarize what I just talked about, routinely collected rapid diagnostic tests, used RDTs can be, and also DBS can be reliable sources for this kind of really fine scale level of analysis, which requires deep sampling. And we observed a spatial trend of highly related and connected parasites. We also think that individual level and really fine scale level analysis will give us better resolution. The genetic networks can show us the scale of local transmission and the length of these local transmission chains, and will also contribute to some extent to determine the contribution of cross-border importation to local transmissions. So I would like to acknowledge my group, the Greenhouse Lab, and Brian Greenhouse for his mentorship, and all the lab people. This study was funded by the Bill and Melinda Gates Foundation. And thank you.
We have time for one question. Is there any questions from the floor? No? Okay. Sofonias, it looks like there's a differential distribution of your population assignments. Does that correlate with the population structure, the fine-scale population structure that you're seeing?
Differences in the sample size, you mean?
No, it looks like there's more population structure in the eastern region. Mm-hmm. So there were more samples that were correctly assigned.
Yeah, I think that's related to the history of transmission in that area. So the western side is more high transmission area than the eastern side.
Okay.
Yeah.
All right. Thank you.
All right, thank you.
Thank you.